Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
For the longest time, when we talked about cancer, it seemed, well, pretty straightforward, didn't it?
Yeah, the basic idea was it's a genetic thing, faulty genes, mutations building up.
Exactly. And that idea, it drove so much research. I mean, mapping cancer genomes, developing targeted drugs, huge efforts.
Absolutely massive. But some of the results from that very research are, well, they're making people rethink things a bit.
It's like the tools we built to confirm the theory are now showing us its limits, maybe?
That's a good way to put it. And that's really the core of a pretty fascinating essay that came out recently.
Oh, yeah.
It's called The End of the Genetic Paradigm of Cancer. Came out in PLOS Biology, March 18th, 2025. The authors are Sui Huang, Anna Soto, and Carlos Sonnenschein.
Right. I think I saw something about that. Wasn't there a SciTechDaily piece, too? The new cancer theory that could change everything we know? Something dramatic like that. Same day, March 18th.
That's the one. It picked up on the essay's main points. It suggests this whole focus we've had on somatic mutations, changes in DNA in regular body cells as the main driver, well, it's facing some serious questions.
Okay, so that's what we're digging into today, then.
Exactly. In this deep dive, we really want to unpack that. Look at why this long-held idea is being challenged.
So we'll look at the data that doesn't quite fit, the inconsistencies.
Yeah, the results from all that genome sequencing that were kind of unexpected. And then explore the alternative ideas popping up.
Ideas beyond just the mutations themselves.
Right. Things like gene regulation, how genes are turned on and off, and the different sort of states a cell can be in, and also the bigger picture, the tissue, the environment around the cells.
And this isn't just theory for theory's sake, is it? The SciTechDaily article hinted this could change, well, everything. Treatments, prevention.
That's the hope. If we shift our thinking, it could open up completely new angles, especially for tackling things linked to, say, environmental factors that don't necessarily work by directly damaging DNA.
Okay, let's set the scene first. The traditional view, this somatic mutation theory. Can you just quickly recap the basics?
Sure. So the core idea is cancer starts in one single cell.
Okay.
That cell gets a genetic mutation, maybe from radiation, a chemical, or just a copying error when it divides.
And this mutation isn't good, obviously.
Right. It gives the cell some kind of advantage. Maybe it grows faster, or it ignores signals telling it to stop growing, or it avoids dying when it should.
Like cellular natural selection, survival of the most messed up cell.
Kind of, yeah. The mutated cell divides, passes the mutation on. Its descendants might pick up more mutations, getting even more advantages.
Yeah.
Eventually, you get a whole clump of these abnormal cells, a tumor, all descended from that original one. Clonal expansion, they call it.
Okay, makes sense. Simple, logical progression. But you're saying the evidence, it doesn't always line up so neatly.
That's what the essay argues. There are quite a few paradoxes or inconsistencies that have emerged from all the sequencing data.
Like what? Give me an example.
Okay, we'll take some really aggressive cancers. You'd expect them to be riddled with obvious driver mutations, right? The specific genetic changes pushing their growth.
Yeah, logically.
But sometimes you look at cancers like, say, rhabdomyosarcoma or certain ependymomas, nasty cancers, but they often lack a clear, consistent set of those expected mutations. It's hard to point to the genetic cause.
Huh. So the engine driving them isn't always the one we thought.
It seems that way sometimes. And then there's the flip side.
Which is?
We find classic cancer-causing mutations, oncogenic mutations, in perfectly normal, healthy tissue all the time.
Wait, really? So cells can have a known cancer mutation, but not be cancerous?
Exactly. Skin, esophagus, blood.
Keep wincing finds these mutations in healthy people, in tissues that never develop cancer. Which poses a big question. If the mutation is the cause, why doesn't it always cause cancer?
It's like having the key, but not turning it in the ignition.
Precisely. It suggests the mutation itself isn't the whole story. Something else must be involved in whether that mutation actually leads to disease. And the essay mentioned something about chemical carcinogens too, didn't it? That the mutation patterns they cause aren't always distinct.
Yeah, that's another puzzle. Sometimes the signature of mutations in a cancer known to be caused by a specific chemical looks pretty much identical to the mutations in a cancer that just seem to arise spontaneously.
Which makes it harder to blame the chemical directly via its mutational effect alone.
It does complicate that direct link, yes. And think about the sheer number of genes implicated in cancer over the years.
It feels like a lot.
Some analyses suggest almost every gene in the genome has been linked to cancer in some study or another, which makes you wonder if we're really talking about specific cancer genes, or if it's a more, system-wide vulnerability.
That's a bit mind-bending. Okay, what about things like moles or ductal carcinoma in situ in the breast? Precancerous stuff?
Right. Those often have well-known driver mutations, classic cancer mutations.
But most of them never become invasive cancer, do they?
The vast majority don't. They just sit there. And sometimes, if an invasive cancer does develop nearby, genetic analysis shows it didn't even arise from those original mutated cells in the lesion.
Seriously. So the initial bad cells weren't the ones that ultimately caused the problem.
Sometimes, yes. It really challenges that simple, linear A to B to C progression model. It suggests maybe the mutation isn't sufficient. The context, the tissue environment must be playing a huge role. Maybe the whole area, the field, is somehow primed.
The essay talked about radiation effects too, right? Not just causing one bad mutation that takes over.
Exactly. After radiation exposure, you often don't see just one single clone expanding. Instead, later on, multiple genetically different groups of cancer cells can pop up.
Suggesting the initial hit wasn't just about one specific gene mutation, maybe? More like general stress or damage?
It could be. Maybe the radiation causes widespread stress or inflammation or tissue injury, and that's the more immediate trigger, rather than a single specific mutation taking hold right away. Genetic instability might come later as a consequence.
Okay, and this idea of positive selection, that cancer cells with the best mutations outcompete others, that's also maybe not the full picture.
Well, you do see selection, but it's often not as clean-cut as we thought. When you look inside a tumor, it's usually not one dominant clone that's taken over completely.
It's more mixed.
Often, yeah. You see incredible genetic diversity, lots of different subclones coexisting. Much of this variation seems to be just random drift, mutations spreading by chance without offering a huge survival advantage.
So it's less like a conquering army and more like a chaotic ecosystem.
That's a good analogy. This heterogeneity, this mix, makes targeting the cancer with drugs aimed at one specific mutation really difficult, because other cells without that mutation can just keep growing.
And it doesn't look like a simple straight line of evolution either, more like a complex branching tree.
Precisely. And then think about cancer coming back after treatment recurrence. When you analyze recurrent tumors, they're often composed of multiple clones again, many of which weren't even detectable in the original tumor. Sometimes the recurrent tumor doesn't even seem genetically related to the primary one in a direct lineage.
Which really messes with the idea that recurrence is just a few resistant cells left behind from the first tumor.
It does. It again points towards the tissue environment, maybe influencing where and how a new cancer process starts, perhaps involving new mutations or activating dormant potential.
Okay, so if the purely genetic story has all these holes in it, what are the alternatives the essay proposes? You mentioned gene regulation and tissue environment. Let's start with the gene regulation bit.
Right. So this view shifts the focus. Instead of looking just at permanent changes in the DNA sequence, the mutations, it looks at how gene activity is controlled dynamically.
How genes are switched on or off.
Exactly. What a cell looks like, what it does, its phenotype, isn't just about the genes it has, but which ones are active and how they interact. This happens through a complex network, the gene regulatory network, or GRN.
Okay, the GRN, like the cell's operating system.
Yeah, think of the genome as the hardware and the GRN as the software deciding which programs run when.
And the essay talks about attractor states, like stable modes for this network.
That's the concept. The GRN, through all its internal feedback loops, naturally tends to settle into stable patterns of gene expression. These are the attractor states.
Like being a liver cell or a skin cell?
Exactly. Those are healthy attractors, corresponding to normal cell types and functions. But the theory is that for any given genome, there are potentially many other stable states the network could adopt, but normally doesn't in healthy tissue.
Uh-huh.
And the idea is that some of these alternative unused states are actually cancer attractors, latent, undesirable states that represent cancerous behavior.
So cancer isn't necessarily caused only by a new mutation creating a new function, but maybe by something disrupting the network and pushing the cell into one of these pre-existing, but usually dormant, cancer programs.
That's the core idea. A disruption, Maybe a mutation, maybe chronic stress, maybe an environmental toxin. Doesn't have to invent cancer from scratch. It just has to knock the cell off its stable, healthy state and into one of these latent cancer attractors.
Wow. That feels quite different. It implies the potential for cancer is kind of built in.
In a way, yes, as a possibility within the system's dynamics. These cancer attractors, because they aren't part of normal development and aren't maintained by evolutionary pressure in the same way healthy states are, might represent more primitive, uncontrolled cell behaviors.
Like reverting to an earlier evolutionary state. Less specialized, more focused on just dividing.
Something like that. And it fits with observations that cancer cells often look de-differentiated. They lose the specialized features of the mature cell type they came from. They seem more like immature or embryonic cells in some ways.
Because those cancer attractor states weren't involved in making the final specialized tissue.
Right. They represent states that could exist based on the genes we have, but which development normally bypasses or suppresses. They might inherently favor proliferation, plasticity, even genetic instability, things useful in early development, but disastrous in adult tissue.
Okay, that's the cell state dynamics idea. What about the other one, the tissue organization field theory? That sounds like it zooms out even further.
It does. It shifts the focus dramatically, actually. Away from the individual cell, mutated or not, and onto the tissue context, the environment, the organization.
So it's saying cancer isn't primarily a disease of a rogue cell, but a disease of tissue organization breaking down.
Pretty much. The argument is that normal tissues actively maintain order. They provide signals, architectural constraints that keep cells behaving properly, suppressing their inherent tendency to just proliferate.
Cells want to proliferate by default.
That's the idea in this theory. Proliferation is the default state, and normal tissue structure and communication actively restrain it. Cancer happens when that restraint breaks down, when the tissue organization is disrupted.
Yeah. So the field goes bad, and that allows cells within, maybe even cells without major driver mutations, to start behaving like cancer cells.
Exactly. The problem originates at the tissue level, not necessarily the cell level. The essay mentions some striking experiments supporting this.
Like the liver cancer cell experiment?
Yeah, that's a key one. They took aggressive liver cancer cells, injected them directly into a rat's liver. They formed tumors, as expected.
Right.
But if they injected the same cancer cells into the spleen first, which then caused the cells to disperse individually throughout the liver and integrate into the normal liver structure.
What happened?
No tumors. The cells behaved normally. They participated in normal liver functions. The healthy tissue environment essentially normalized the cancer cells.
That's incredible. It suggests that cancer's behavior wasn't locked into the cell's genes, but was dependent on its surroundings.
It strongly supports the idea that the tissue context is paramount. It's not just about what the cell is, genetically, but where it is and what signals it's receiving.
And this theory also puts a lot of emphasis on the stroma, right? The supporting tissue.
Absolutely. The stroma, the connective tissue, blood vessels, immune cells, the extracellular matrix isn't just passive scaffolding. The TOFT theory sees it as a key player, even a primary target.
How so?
Changes in the stroma, maybe due to inflammation, injury, or carcinogen exposure, can disrupt the normal tissue organization and signaling, thereby causing nearby epithelial cells, the main functional cells, to become cancerous.
So the damage might be to the support structure, which then leads the main cells astray.
Precisely. There were experiments mentioned where they exposed only the mammary stroma in rats to a carcinogen, shielding the actual milk-producing epithelial cells. Mammary cancers developed from the unexposed epithelial cells. But if they exposed epithelial cells to the carcinogen in a dish and then transplanted them into a healthy, untreated stroma, they formed normal structures.
Wow. So the state of the stroma, the soil, determined whether the seeds grew into cancer.
It's a powerful demonstration of the stroma's influence. And it goes beyond chemical factors, too. Even physical things matter.
Like what?
Like just implanting a solid, chemically inert piece of plastic under the skin can cause tumors. But injecting the same material as a powder doesn't.
Why?
The solid object causes chronic inflammation, fibrosis, mechanical stress. It disrupts the tissue architecture. The powder doesn't cause the same kind of long-term physical disruption. It suggests physical forces and tissue structure are part of the control system.
Okay, this is a lot to take in. It really shifts the perspective from just hunting for mutations.
It certainly broadens the view significantly.
So if cancer isn't just about genes, if it's also deeply tied to gene regulation, cell states, tissue organization, What are the big takeaways here? What does this mean for research, prevention, treatment? The essay called for deep thinking alongside deep sequencing.
I think the main implication is that we need a more integrated approach. We can't just focus on the genetic mutations, as important as they are. We also need to understand these other layers, the regulatory networks, the tissue context.
Which means looking beyond DNA sequencing.
Yes, it means seriously investigating non-genetic factors. Environmental exposures, lifestyle, chronic inflammation, maybe even things like tissue mechanics. How do these things disrupt normal regulation and organization?
And for treatment, if the tissue environment is crucial,
Then maybe therapies need to target the environment, not just the cancer cells themselves. Therapies that restore normal tissue structure or communication, or target the problematic stroma. It opens up different avenues.
and it fits with that updated quote from Mike Stratton, wasn't it? A leader in cancer genomics, suggesting chemicals might cause cancer in ways other than direct mutation.
Exactly. It signals a potential shift even among those heavily invested in the genetic view, a recognition that the mechanisms might be broader, involving these regulatory and tissue-level effects. We need to understand how these non-mutagenic factors might push cells into those cancer attractor states or disrupt tissue fields.
So it's about understanding the system's dynamics, the loss of normal stability, not just identifying the broken parts.
Precisely. The essay argues for embracing formal theory, thinking about biology's inherent complexity and history, not just cataloging molecular parts. Perhaps the relentless search for biomarkers sometimes overshadowed understanding the fundamental biology.
It definitely leaves you with a lot to chew on, that final thought. If cancer isn't solely a genetic disease, what does that mean for us? For how we approach preventing it, finding it early, treating it?
It forces you to consider the whole picture, doesn't it? The intricate dance between our genes, yes, but also our environment, our lifestyle, the very way our tissues are built to maintain. It pushes us to ask, beyond the mutations, what else is driving this incredibly complex disease, and what new strategies can we develop based on that broader understanding?
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.